Hello, everyone. Thank you for coming to today's endocrine seminar. And our speaker today is Dr. Banrida Wahlang. And Banrida is an assistant professor in the Department of Pharmacology and Toxicology. And she is a real rising star in the field of environmental exposures and sex differences. And you'll see why. So Banrida received her BS in pharmacy from the University of Delhi in 2008, and then a Master of Science in Pharmaceutics from the National Institute of Pharmaceutical Education and Research in Najjar, India. She came to UofL in 2010 and completed her Ph.D. in 4 years in pharmacology and toxicology in the lab of Matt Cave. And during that time, she contributed to 7 papers on which she was Forrest's first author. And this is the kind of person that Banrida is. Overall, I think she has 54 publications, many of which are first author. Following completion of her PhD here at UofL, she traveled down the road on 64 East to the University of Kentucky, where she spent 2 years in postdoctoral training in the lab of Dr. Bernard Henning, who has great expertise in PCB, polychlorinated biphenyls. And there she was an author on 5 papers and 2 as first author. Then she came back to UofL as a postdoctoral associate and NRSA fellow in the UofL Alcohol Research and Superfund Research Centers. And during that time, she published 18 papers with 6 as first author. Then she became an instructor in medicine for a year, and she continued to collaborate with Matt Cave and with Craig McLean and with my lab during that time. And she provided expert mentoring in mouse work and PCB administration to Dr. Belinda Petrie, who is right here, and Kelly Peel, who is upstairs, working in my lab. July 1st, 2022, in 2022, she became a tenure-track assistant professor, and she is well on her way to success. She's had 10 papers during the time she's been an assistant professor. She's clearly very organized and hardworking and has demonstrated outstanding collaborative skills. She's currently PI on an NIH KO1 from NIEHS, evaluating mechanisms on sex differences in an environmentally induced metabolic diseases. And she's also PI on a DOD TERP, which is Toxic Exposure Research Program grant, acute and subchronic exposures to dioxin and dioxin-like compounds in liver disease, the role of sex and age. And she has received CIEHS and UofL core research vouchers to support her innovative research, giving her the tools to be successful on an NIH R01, for which she just got a really excellent score. And we're hoping for an award notification soon, I think, for Banrida. Banrida has also contributed in many ways to the university as in service. She's currently involved in the CIHS P30 Center as a leader of the career development program for junior faculty. And I know from sitting in on those sections, she has excellent mentoring skills, and she's also mentored undergraduate and medical students, and she's currently mentoring 2 PhD students. She's also been involved in service nationally as an NIH and DOD TERP grant reviewer, as an editorial board member of the Journal of Nutritional Biochemistry. And of course, she also does many ad hoc manuscript reviews, including for molecular cellular endocrinology, which I am also grateful. So it's a delight to have Banrida share her research with us today. Thank you, Banrida.
Wow, that was such a nice introduction. Thank you so much, Carrie.
Yeah.
So today we'll be talking about pollution and endocrine metabolic disorders, Battle of the Sexes. I know this is a great, excellent movie, but we'll not be watching this for the next hour. You'll be listening to my talk instead. So there are no relevant disclosures that I need to announce regarding this research today, but I am a member of several centers, just like Carrie had alluded to, and as well as I want to acknowledge collaborators at the NIEHS and the CDC that work with me in my research. So learning objectives for today, well, the first thing is we want to know or identify what environmental toxicants are and how they act as endocrine disrupting chemicals, describe their contributions to liver and metabolic dysfunction, and also understand how biological sex acts as the mediator in disease susceptibility. All right, so let's talk a little bit about environmental endocrinology. So when you think about environmental endocrinology, endocrine disrupting chemicals come to mind, right? And these are basically exogenous chemicals or a mixture of chemicals, if you will, that can interfere with any aspect of hormone action in the body. And when we think about endocrine disrupting chemicals, we only think about it in terms of the chemical, right? As the chemist, like the chemical, the compound. But there is so much other complexity that adds to environmental endocrinology and how these chemicals impact the endocrine system. And these include the physical environment. So for instance, use of cell phones, for instance, could have an impact on our endocrine system, or the kind of water that we drink from our bottles could impact our endocrine system. Chemical environment, like I said, exposure to occupational or environmental chemicals, or residing near a chemical plant, and that puts you at risk for chemical exposures. And then biological environment, so what our bodies, our biology, right? And so sex is a part of that biological environment. Pre-existing health conditions that could impact the endocrine system also influence and shape the impact of endocrine disrupting chemicals, as well as sociocultural environment. And this is really important because our lifestyle factors are highly influenced by our social environment, right? If you go, and vape, or alcohol drinking, or going to work out, or join a Zumba class, all those sociocultural factors also play a role in how endocrine disrupting chemicals can impact the endocrine system. So we always try to look at it from a holistic approach when we talk about endocrine disrupting chemicals. And so to add more to that complexity, these different environments, physical, chemical, biological, and social cultural environment, also interact with each other. And so why does it matter? So why does environmental endocrinology matters, right? So this is a diabetes prevalence map from, I'm trying to use the mouse. Okay, so a diabetes prevalence map from 2021. It was published last year in Lancet, and it shows how the prevalence of diabetes is worldwide. So when I was looking at this map the other day, I was thinking about, how these common risk factors for diabetes, type 2 diabetes, if you will, like obesity or high caloric diets, they don't really add up, because you look at some of the diabetes prevalence, especially in the sub-Saharan African region, or even in the Middle East, or even in Southeast Asia, if you will, those are traditionally regions where you don't really see a lot of that Western diet effect, right? So this is more of that Eastern diet effect. But recently, with more processed food coming into play, and more high sugar, high calorie foods that these countries can have access to, there has been a surge in these metabolic dysfunction, metabolic diseases. And another aspect of it is also, which we, especially this paper also didn't talk about it, but especially important for us, is what about environmental chemical exposure that could be present in those processed foods, for instance, like PFAS chemicals that have been found to be in food wrappers from McDonald's, for instance. What about those daily low-level exposures to these chemicals? Could those be a contributing factor to these trends that we're seeing, other than these demographic shifts in high caloric intake or processed food intake? So we don't know that yet, and that's why it matters, because our research matters for that reason. And so with that in mind, our research objectives are to look at sex differences in environmental health and metabolic health and liver disease. And our endpoints that we look at are endpoints of the metabolic syndrome. We look at cirrhotic liver disease, and to an extent, we've been starting to indulge ourselves in cholestatic liver disease with environmental chemical exposure. And so our environmental contaminants of interest are persistent organic pollutants. For those of you that don't really know what they are, I'll be talking at length about them today. And the second clause of volatile organic compounds, I'm not going to talk too much about those today for the sake of time. And what we're trying to do here is to understand what are these sex-specific mechanisms of these environmental chemicals and how they induce toxicity. So when I was first interested in sex differences in environmental health, people would say, so what? You see differences in males and females, so what? But the so what for me is, what if those mechanisms that contribute to those toxicities are different, right? And so that could shed light in who is at risk, the population that are at risk. And it could also shed light into intervention strategies that we could use that could be sex specific. Maybe we missed it in our male models, but maybe those could be effective in our female models. And we generally use a translational or a reverse translational approach. We draw observations from a group and population studies, take that information, go back to the lab, and then we do experimental models or mouse models of experimental exposures to see or discriminate those mechanisms of toxicities. And so let's talk a little bit about POPs now. So persistent organic pollutants, or POPs, are my favorite chemicals, unfortunately. They're also known as forever chemicals because they persist in our environment. Unfortunately, POPs are man-made. We made them. Humankind is responsible for them, for being present in our environment and our ecosystem today. And they were produced for industrial purposes. And sometimes they even were produced inadvertently, unintentionally, as byproducts of industrial processes or activities. They are persistent in our environment, and a reason for that is because these chemicals, they're usually highly chlorinated or highly fluorinated, and so they're resistant to degradation. They're resistant to P450 metabolism in our body, and they tend to bioaccumulate in adipose tissue of living organisms because they're also very, very hydrophilic, lipophilic, so they're fat-loving. And so if you look at this map over here that I wanted to show you, so if you look at these POPs that were listed under the Stockholm Convention, this was a convention that first met in Europe in 2001 when a bunch of countries figured out that, okay, these persistent organic pollutants are bad for environment, they're bad for our human health, and they're bad for our ecosystem. So how do we stop and how do we regulate them? So they met in 2004 where they started this whole, they called it a dirty dozen list, where they put 12 chemicals where they restricted their use or restricted their production. And so in that dirty dozen list, there were chemicals like organochlorine pesticides, like DDT and chlordane, and then there were PCBs, polychlorinated biphenyls, and there were dioxins as well. But as they kept meeting over the years, this list under the Stockholm Convention started increasing. And in fact, in 2010, 2011, they put PFOA, which is one of the popular forever chemicals that's in the news today. It's everywhere. I just watched the news yesterday in Spectrum, and it was like forever chemicals are found in animal tissue, so please don't go hunting and eat those deer that you were hunting or something like that. And so they put PFOA under the list in 2010, but guess what? Now we're like 14 years later and we're still having issues because a problem with these chemicals is not these chemicals, but with companies companies or industry that make these chemicals was, if one chemical was banned, then they just changed the structural moiety of the compound, introduced that into the market because it's not regulated, even though it may do the exact same thing as that parent chemical to our bodies. And they still would say that, okay, we're going to make this instead. And then it was just, a vicious cycle of getting these chemicals regulated. So another problem when it comes to these forever chemicals in our world today is also because of the issue of climate change. So with climate change and changes in temperatures in our environment and our globe and our planet globally, what happened is that these POPs could migrate from regions of low latitudes to regions of high latitudes. And so, for example, if there was an organochlorine pesticide that's not used anymore in the States, like DDT, for instance, I know that about still a decade ago, it was still used in developing countries like India and China. And so those levels of the DDT still persist in those countries. And with global warming, we have the possibility of those chemicals being transmitted from the regions where they're being used to regions where they have been banned and restricted. And so there is this issue of migration of these chemicals, and it's also known as the grasshopping effect. And another important thing about these persistent organic chemicals to remember is that they occur in the environment as mixtures, and they're often detected in combination when it comes to soil, water, and air samples. So if you were taking fish from the Ohio, send it to a lab, and ask them to measure PCBs and PFAS, they will come back and actually show you that, concentrations of PCBs and PFAS. And I know this because I just reviewed one of those reports 2 days ago. So there's still very much an issue. All right, so metabolic diseases. So like I said, we were interested in looking at endpoints related to the metabolic syndrome, such as obesity, diabetes, blood glucose levels, dyslipidemia, and to an extent, we haven't really looked at hypertension. We didn't have the means yet, but I hope that as we grow our lab and as we grow our collaborations, we'll also be able to look more into the heart-liver axis. But for now, we're focusing more on diabetes and steatotic liver disease, which is the liver manifestation of the metabolic syndrome. So when we talk about sex differences or, sexual dimorphic prevalence of these diseases. When it comes to type 2 diabetes, males are usually more susceptible. That's kind of, widely known, even if they have lower BMI thresholds. And in fact, the prevalence, the male to female ratio for type 2 diabetes is 1:14. But I want to tell you that there are actually a handful of countries, about 60 countries in the globe where the prevalence is reversed. So actually females are more susceptible to diabetes, or the prevalence, if you will, is more in females than males. And that was blamed on, females being having a more sedentary lifestyle than males and being more obese, if you will. So when it comes to steatotic liver disease, which is this progression of disease from a normal liver to a steatotic liver, which is 5% or more fat accumulation in the liver, to steatohepatitis, which is basically steatosis accompanied with inflammation, which can then progress to cirrhosis and even hepatocellular carcinoma. When it comes to that, the prevalence could be different looking at the cause. So if you look at metabolic dysfunction associated steatotic liver disease or MASLD, this is often blamed on Western diet, hypercaloric diets, and lack of exercise, if you will. Females are less, so premenopausal females actually appear to be more protected against MASLD. And it appears to be more prevalent in males and postmenopausal females, suggesting that perhaps, high levels of estrogen in our bodies could be a protective mechanism for that. But if you look at alcohol-associated liver disease, it seems to be more prevalent in females. And this was not the case even like 5 years ago, because there was not enough research done in the female population when it comes to alcohol-associated liver disease. It was always thought to be more prevalent in males. But now with this knowledge gap, narrowing down, we're getting more information on these sex differences and disease prevalence. And so what about toxicant-associated steatotic liver disease, which is the steatotic liver disease that we're interested in, that we know that could be caused from exposure to environmental or industrial chemicals? Well, we don't know yet about sex differences. And so that's why, again, our research is highly relevant and could shed light into this field, if you will. So I want to touch base again a little bit on why we are interested in sex and gender in environmental health. So we are exposed to environmental chemicals throughout our lifespan, and throughout our lifespan, we may encounter different, elements, different changes or events that could modify biologically, that could have an effect on our biology. And these could include things like pregnancy or lactation or any other kind of pre-existing condition, if you will. And so it's highly important to consider these factors when we're assessing risk for these environmental chemical exposure and how they differ, especially in terms of biological sex. So factors that influence sex differences, it's easy, right? It's like, okay, it's the XY versus XX chromosome. The SRY gene in males that contributes to those hormonal, organizational, and activational effects. But also there are also non-gonadal effects like the differences on unequal expression of X and Y genes that could cause the sex chromosome effects that could contribute to sex differences in organ systems or even in our response to xenobiotics and so on. And so I like to look at this from 4 standpoints. So one is genetic standpoint, one is phenotypic standpoint, and then one is hormone levels. What are the effects on hormone levels, as well as reproductive elements? So you kind of take, when you look at chemical exposure and risk for metabolic diseases, kind of take all those standpoints into consideration. And how does gender come into play as well? And this is a classic example that I like to give everybody when I have the sex and gender spiel. So why does it matter in terms of environmental health sciences, right? So when we think about biological sex, so things like differences in skin thickness could contribute to, differences in how a chemical, for instance, can enter a body to dermal absorption, how much of that chemical could enter a body. But when we think about gender is a sociocultural identity, right? And it defines us and what we do and how we do. For instance, for a woman, for a person or individual that identifies as a woman, they may tend to use chemicals or beauty products that men don't often use. And so that could influence the type and the amount of chemical, or what kind of chemical is that person exposed to versus the other gendered person. So that's why it's really important to kind of look at this together from a sex and gender standpoint. Unfortunately, I work with mice, and mice don't really have a gender identity, so we're kind of stuck. But hopefully into the future, if I could find people that are interested in this kind of research, we can look at, for instance, in patients or in population studies as well to look at this integration of sex and gender and how that impact environmental toxicity. All right, so I do have some ongoing research to show you that why should we care about these sex-specific environmental effects. So I want to share with you some preliminary data from this Anniston Community Health Survey study, which is a population that live in Anniston, Alabama. So Anniston is a really small town in Alabama that was home to the Monsanto plant that manufactured PCBs back in the day, but it also is near multiple military bases, as well as had plants that manufactured organochlorine pesticides. So this population have levels of persistent organic pollutants, including PCBs, PFAS, chemicals, at really high concentrations, at really high levels, even 3 times or more than the normal general American population like you and I in this room that are hopefully not exposed to these chemicals to that extent. And so first thing we did was let's see if these chemical levels are different in our males and females. So we decided to go ahead and look at these levels. And what we saw was that we started with our PCBs because we're interested in these PCB chemicals. And we found that in both our initial cohort as well as a follow-up cohort, females tend to have higher circulating levels of PCBs and even organochlorine pesticides than our males. And we're like, great, so we're seeing these differences in levels. What does that mean? So we went ahead and did some preliminary sex-specific associations between these chemicals and liver disease biomarkers. And what we found was that, I'm going to show you 2 over here. I'm not sure if you're able to see it, but this is an oxychlordane. It's a metabolite of chlordane, which is a organochlorine pesticide. And our females are in blue and our males are in dotted reds here. So what we saw was that oxychlordane serum levels was positively associated with M65, which is a liver cell fat marker, only in our females, but not in our males. And the same thing was observed for interleukin-6 or IL-6, which is a pro-inflammatory cytokine. So we were like, yay, we see some sex-specific associations, and that's great. So, yes.
Were these premenopausal women or postmenopausal, or a mixture of ages?
They are a mixture. But I believe the age was about between 40 and 50. So not really, kind of like on the cusp. So yeah, but that's a good question. Okay, so another thing that we look at was another population study. So this was the HEAL study, which is the Health, Environment, and Action in Louisville study. This population was from the southern Louisville area, and we were looking at totally different chemicals. So we were not looking at the forever chemicals or like PCBs. We were looking at VOCs, so volatile organic compounds. And I'll just walk you through this. But what we basically saw was that we stratified this population by males and females, and we measured 16 urinary VOC metabolites as exposure biomarkers. And our disease biomarkers were, liver enzymes like ALT, AST, and ALP, as well as bilirubin, and our cell death markers like M65. And so we did correlation analyses. We also stratified it by race. We wanted to look how racial differences impact chemical association with liver disease biomarkers. But what we really found here was that biological sex really played a role in these associations. So we only found positive associations between these, about 8 of these metabolites with the liver enzyme ALP only in females. And, strikingly enough, it's not just females that don't smoke, it has to be females that smoke. So if you're a female who is a smoker, then you're more at risk for these elevated liver enzymes with VOC exposure. But we never saw any of these effects in our males, surprisingly. And the funny thing is that females generally tend to have lower levels of circulating liver enzymes anyway. So we tend to try to make sure that there's no confounders. So we really are we really are confident in these results because we really did a mixed, so we combined both on males and females. Again, we did see some associations only in smokers, but again, when we stratified it, we only see this only in our females. And so this was one of the things that pushed us into looking at cholestatic liver disease, because ALP, alkaline phosphatase, is a prognostic marker for cholestatic liver disease as well as bone disease. And there were some epi studies at the time, and I was working on the manuscript for this paper, that I read that women that work at nail salons, for instance, that are, they have a higher risk of being exposed to volatile organic compounds like acetone and such. They tend to have increased elevated levels for ALP as well. So this is a different kind of line of research, but we would be interested into pursuing that as well. All right, so enough about population studies. Let's get to my studies, okay? So another thing that we look at in our lab is to study multi-organ toxicity. And the reason for this is because, again, when you think about these metabolic complications like obesity and cirrhotic liver disease, generally the conventional thinking had always been that, the energy input and the energy output doesn't match each other, but also could be genetics and nutritional habits. But now we are, and that could lead to obesity, diabetes, and metabolic dysfunction, cirrhotic liver disease. But now we know that lifestyle factors play an important role: what we eat, what we drink, what we smoke, and how much of that are we doing. And then you add to that environmental factors like chemicals or environmental stressors, if you will, and all that could play a role to impact multi-organs simultaneously, leading to organ-organ crosstalk or organ-organ interactions, eventually impacting obesity, diabetes, as well as cirrhotic liver disease. And so with this in mind, this theory in mind, our hypothesis is that, exposure to environmental chemicals like POPs, for instance, along with the added factor of sex differences could play a sex-specific role, or could lead to sex-specific phenotypic changes in the liver and the endocrine system, as well as in the gut, because these are major organs that actually could crosstalk with each other to lead to metabolic dysfunction. So we come up with this endocrine gut liver axis, if you will, and now 2 years down the road, we've realized that it's more complicated, because then, if you look at, I get it why people always like to focus on one organ, it's just easier. And because you have so many other factors to think of doing multiple organs assessment. And so I'm going to show you one of our toxicological studies that we did, and what the effects are in terms of sex differences, and what we think are the mechanisms for those sex-specific differences. So we did this 12-week animal exposure study where we had male and female Black 6 mice that were early adult, about 6 to 8 weeks of age, that were eaten on a Western diet, which is a 40% fat diet, or a low-fat diet, which is about 10% fat diet. And we exposed these animals to a mixture, because we are into mixtures, right? Because these chemicals are found in our environment as mixtures, it's relevant to study them as mixtures rather than individual components. And so we exposed them to a mixture of chlordane, which is an organochlorine pesticide that has non-dioxin-like properties, and PCB126, which is a dioxin-like chemical. And so the reason for doing the specific mixture is because these chemicals activate different receptors. We want to assess how simultaneous activations of those receptors impact metabolic outcomes. And we did a 12-week study. At the end of the study, we collected a bunch of tissue and blood and did a number of analysis. So I'm going to show you what we saw. So we did a glucose tolerance test, obviously before the mice were euthanized. I believe this was towards the end of the study period. And so we saw that, surprisingly, or not surprisingly, our male mice seem to be more resistant to glucose uptake with exposure than our female mice. So our greens are our males, our purples are our females. So you could see this area under the curve which reflects blood glucose levels with time, that it was actually higher in our male exposed compared to their sex match controls, whereas in our females, they don't really have a significantly different area under the curve than their sex match controls. And we said, okay, this falls in line with the general paradigm that males are more susceptible to, let's say, type 2 diabetes and glucose intolerance. So we also put these animals in metabolic cages to look at energy expenditure and physical movement. So we didn't really see much differences with food intake or energy expenditure, but we saw was that in terms of activity, while our females are generally active, our female exposed mice tend to be more lazy and just hang out and sit and observe rather than move around. And so we saw this phenotypic difference, if you will, in movement or activity. And we thought, yay, we are seeing some kind of phenotype here. And so let's go and look at the liver and what happens there. So we went ahead and looked at our liver sections when the mice have been euthanized and livers have been processed. And what we found was that if you look at these top panel here, these are on males, so this is with control and with exposed. So this is with the high-fat diet. I'm going to show you just the Western diet. I'm going to show you just the Western diet findings right now. And so the males tend to have more both micro and macro vesicular steatosis, which is more diffused. And if you look at our females over here, they tend to have less steatosis to begin with, even though they were on the same diet with the same fat content. And they generally have more of a zonal macrovesicular steatosis if they have it at all. And so we also did a all red oat staining to confirm those H&E staining that we saw, and we also quantified hepatic lipids. And what did we see here? So if you look at our hepatic triglycerides and our hepatic cholesterol levels here, so in concordance with our histology findings, males tend to have more steatosis than our females, and this is more hepatic triglycerides in our females, and this is something we've seen in our studies routinely. It's because female black 6 mice are more resistant to hepatic lipid accumulations than males. Especially on a high fat or a Western diet content. But if you look at our females with exposure, they tend to have higher hepatic triglyceride and cholesterol levels than our sex match control. So it might not be a big difference, but it's there, and we were excited about that because we actually saw a sex-specific difference, although we don't know what to make of it yet at the time. And so we also look at inflammation and liver function. So we measured, a bunch of hepatic genes encoding cytokines and fibrotic markers. We only saw sex differences, so nothing exciting there. But we did see that with hepatic albumin expression, we saw that there was a decrease in our female exposed mice in terms of hepatic albumin expression compared to their sex match controls. But we didn't see that in our males. So the female mice tend to be more susceptible, if you will, to steatosis, as well as there seems to be some kind of hint towards, liver dysfunction. And we are still haven't measured protein levels yet or circulating albumin yet. But I just want to add here that in our ACHS study, we did see a similar trend. We haven't done it by sex, but we saw that in both our male plus female population, that especially with the dioxin-like chemicals, the dioxin-like PCBs, there was an association with decreased circulating albumin levels. All right, So we also looked at antioxidant capacity, measured hepatic glutathione levels in the blood as well as in the liver. We didn't really see differences in the blood, but again, in our liver, we saw that in contrast to what we saw with hepatic steatosis, actually male-exposed mice seem to have lower hepatic GSH levels, as well as the GSSG level, which is the reduced glutathione to oxidized glutathione levels, indicating that there's some kind of antioxidant capacity that has been compromised in our males, but not necessarily our females. Okay. We also, so before we go ahead, we wanted to look at mechanisms, right? So we know that in terms of receptor activations, so persistent organic pollutants are xenobiotics, just like drugs, they could go and activate xenobiotic receptors. And one of the pronounced xenobiotic receptors that people have looked at, for several decades now, if you will, is the aryl hydrocarbon receptor, or AHR. And this is a target for more of the dioxin-like chemicals like PCB126, which is one component of the mixture. And other xenobiotic receptors that these chemicals could act is constitutive androstenedione receptor, or CAR, as well as others. But I'm going to focus on these two because those are the two primary receptors that this POP mixture that we use act upon. And so we did a liver proteomic analysis on these high-fat these high-fat content liver of these mice, and we saw that strikingly the proteins that were upregulated in both sexes were CAR and CAR and AHR target genes, such as CYP1A1 and CYP2B10. We didn't really see the CYP3A11, which is a target gene for PXR. We didn't really see that upregulated. So we know that the AHR and the CAR were the ones that were at play here. Surprisingly, we also were able to look at sex exposure interaction effects, and we saw that fatty acid synthase of FAS protein levels were up in our we noticed that there was a sex exposure interaction effect. So we went back to the lab and we did a bunch of Western blots to first validate our proteomic findings. And we did see that, CYP1A1, which is the AHR target, was upregulated even in our Western blots, and CYP2B10 was also upregulated. And in our high-fat diet mice, we didn't really see any differences in terms of the levels of upregulation of these targets. And we also saw, interestingly, that fatty acid synthase was also upregulated in our fatty acid synthase proteins was upregulated in our female exposed mice compared to their sex match controls, which perhaps could explain some of those hepatic steatosis that we saw in that group. And so are females more susceptible to steatosis then? So we were fortunate enough to do another study at a time in the lab where we looked specifically only at dioxin-like chemicals. So we forgot about, yes.
Can we go back? I'm just curious, did you look at VLDL?
We did. I don't believe we saw any differences. I mean, there was sex differences, but not with exposure.
Okay.
Yes, but dioxin-like dioxin-like compounds have been shown to suppress VLDL levels in males. Yeah. Okay, so we were doing another study looking at dioxin-like mixtures. It was a short-term study, and this was part of our DoD grant research that we were doing. And so we did the same things, right? We exposed our mice this was a shorter period of time in the absence of the Western diet. This is just a low-fat diet. And what we saw was, again, we saw those same trends in terms of hepatic triglyceride and hepatic cholesterol, especially with hepatic cholesterol. It was, the p-value was even lower, showing that there was a really significant difference. And so this was work done by my graduate student, Esther. And so she went ahead and looked at a bunch of lipid metabolism genes and came back and said, I saw differences in these genes. So fatty acid synthase, SCD1, steroid coenzyme desaturase 1, as well as acyl carboxylase A, and they were all upregulated in our female exposed animals compared to their sex match controls, but not in our males. So our next question was, is this an AHR driven effect where we see this, increase in these de novo lipogenic genes? And so traditionally, the aryl hydrocarbon receptor, for those of you who know xenobiotic detoxification, I'm probably preaching to the choir, but you know that if it hangs out in the cytosol, it's bound by heat shock proteins, etc. And then when a ligand such as a dioxin like PCB or PCB126 or TCDD enters, binds to the receptor, it separates from its scaffolding proteins and then moves to the nucleus and goes sits after heterodimerization, sits in a dioxin response element to promote the gene expression of its target, like CYP1A1, etc. But AHR, there's been a study or a few studies from very reputed AHR-centric groups that have also looked at how AHR, independent of the dioxin response elements, could behave in terms of lipid metabolism. And so one of these studies have looked at AHR-SREBP interactions where they hypothesized that in the cytosol, AHR is hanging out with SREBP, suppressing it from going into the nucleus to do its job. And so what happened is that we think that in our study that when the dioxin-like chemicals come in, they bind to the AHR. What happens is that it goes to the nucleus, and then the SREBP can go to the nucleus and do its job in turning on the de novo lipogenic genes and turning on de novo lipogenesis, eventually subsequently leading to triglyceride synthesis, and so on. And so that's why we're seeing those effects that we saw in our females that were exposed to the dioxin-like mixture. And we also saw that there was a suppression in ESR1, hepatic ESR1 gene expression in the liver for these dioxin-like exposed female mice. But I will not, so there is this whole story about this AHR, ER alpha interactions, but I will not go into that detail today, but I'll focus on another mechanism. And so, all right, so we'll not talk about the AHR, ER alpha crosstalk. So we'll talk about something else. So this was more interesting to us because we actually could tell what in our animal models, we had some significant changes that could answer to that story. So this is the CAR ER alpha interaction. So in 2020, there was this great paper that was published by Nagishi and his group that looked at interaction between the constitutive androgen receptor, or CAR, and estrogen receptor alpha. And they had this mechanism where if you have diabetes or you're exposed to phenobarbital, which is a CAR activator, CAR gets activated, undergoes conformational changes, moves to the nucleus, and after its interactions with ER alpha, conformational changes, phosphorylation, it then sits in the promoter region of SALT21, which is the estrogen sulfotransferase, which metabolizes estrogen and activates it, and then you have lower estrogen levels. And so they had this beautiful paper that talked about it. So we saw that paper and we're like, let's go look at SALT21 levels in our mice. So before we did that, though, we first looked at our CAR activation, which is measured by its target gene hepatic expression, which is CYP2B10, and this was in the low-fat diet group. Okay, so what we saw was that with CAR activate or with exposure to that POP mixture, our female mice and our male mice both showed hepatic CYP2B10 induction, but this was more pronounced in our exposed females when you compare it to our exposed males. So they seem to show more pronounced CAR activation, if you will, if you look at, measure it by hepatic CYP2B10 expression. And so we went ahead and looked at SALT21 levels. Sure enough, we saw that there was an upregulation in SALT21 levels, in our expression levels, in our exposed females, but not our males. And so we also measured plasma estradiol levels, which was a pain in the neck because we used to use a different method and they don't have that available now. So we have to use a huge amount of blood for this, unfortunately, but we were able to look at our exposed groups. So if you look at our female exposed mice, they tend to actually have lower estradiol levels than even our male mice, right? So we have this nice mechanism in place. So we were excited about that, but unfortunately, like every research findings go, we also saw things that we didn't really understand. So when we look at plasma testosterone here, we actually saw that it was upregulated in our males. So perhaps this suggests that this POP mixture is more androgenic and anti-estrogenic, if you will. But we have seen this increase in plasma testosterone in our other POP exposed males in the past. We really brush it off at the time, but we've seen this happening again and again. So I think we need to look more into this CAR, mediated testosterone increase in our male mice, which we don't know at the moment what it is. And so we have that for now. And so what about our OVX model, right? So now we have this nice hypothesis. Okay, these POPs, they can activate the AHR, they can activate CAR, which can lead to this, suppression of ER estrogen receptor signaling or estrogen signaling, if you will, and these could lead to these changes in hepatic steatosis and glucose homeostasis. So we were like, okay, let's test the mechanisms then. Let's test if this is really a true, a female effect. So we did we overreactomized our mice, and then we exposed them to the same POP mixture, but we did this for a 2-week study because we had a grant submission due and we can't wait for 12 weeks to do the study. So we only did it for 2 weeks, unfortunately. And so we did see that effect. So if you look at our sham mice over here that are exposed versus our control, there was that, the area under the curve was significantly higher, but you kind of lose that effect in our OVX mice. So because one of the reasons could be that that mechanism that we thought was in play was actually not in play because these were OVX mice. And we also saw that in our sham operated mice that are exposed to our chemical mixture, there was an increase in liver weight indicative of hepatomegaly, but not necessarily in our OVX mice. Although in general, OVX mice do tend to have, because they don't have circulating estrogen, they do tend to have more hepatic steatosis with time. And so we also look at other endpoints. We look at fat composition. Again, our OVX mice had, higher fat composition than our controls, but our sham controls, but we didn't really see any exposure effect. And part of that could also be because, again, this was a 2-week study, but we also saw surprisingly that hepatic cholesterol with exposure was also increased in our OVX mice. And so, again, like I said, sometimes when we look at our research findings, we, scratch our heads because some things line up perfectly and others don't. So we need to also figure out that, but we haven't done that yet. And so anyways, I'm just going to stop there because I think we are now at 45 minutes. And I just want to talk to you a little bit more about our ongoing research. So we talk about, I showed to you, right, these sex-dependent physiological associations with chemical exposure that seem to be more pronounced with a diet effect, right? Like high calorie diet, high fat diet, if you will. And so, but we were exposed to these chemicals involuntarily from the day we were born, right? Unfortunately. And so so the chemical kind of acts as the first hit. And then what I've shown you that the diet plus a chemical together was working to cause this effect. So now we're thinking perhaps, what if we have a normal liver and it's exposed to these chemicals for a certain period of time, and then we introduce a diet as a second hit, or even another lifestyle factor like alcohol consumption or smoking, or vaping, or a different kind of pollutant, if you will. So with that, push that compromised liver that was compromised because of that first hit, could it push that liver to have toxicant-associated metabolic dysfunction, and what are the sex differences in terms of that? So our new exposure study will be using this kind of timeline where we actually have mice, we'll expose them to the chemical for 12 weeks, and then we'll introduce a high-fat diet or a Western diet, or even alcohol feeding. And then we're going to look for, we're going to do a subchronic study where we look for after another 12 weeks, and we're going to look at what those metabolic outcomes are. And then some of our ongoing research and future studies will also utilize humanized models. So there are species differences in the aryl hydrocarbon receptor that are well known. So we now want to look at humanized receptor to see, are we seeing these effects, and would these effects be, reflected with human exposure? The pancreas is an important organ for us, which we don't have enough expertise. And I'm hoping, talking to you all, that it would open doors for collaborations. We want to assess pancreatic toxicity with these chemicals. We've done a little bit of this in our male models and just a tad in our female models. And we have seen that these chemicals do impact gene expression in the pancreas, as well as insulin, levels or insulin production. We also want to look into gut-liver interactions. We have some data for that, but I didn't show that to you today. But also, what are, there's hope. What are the intervention or therapeutic strategies that we can utilize now that we know that there's some differences in mechanisms for our males and females? And so one way for these intervention studies is to actually manipulate the aryl hydrocarbon receptor, because the liver AHR and the intestinal AHR do completely different things. And we have sex differences in ileal AHR versus liver AHR activation. I haven't shown that data yet, so that could be one way forward. And with that, I would just like to conclude the talk, shown to you that sex differences exist in environmental liver and metabolic diseases. And some mechanisms that could be anti-estrogenic and pro-androgenic. And also, going forward, it's also important to identify, at least in our populations, what are these metabolic and microbial signatures, if you will, that could give us information in terms of disease susceptibility with exposure. And importantly, another thing that, down the road that I'd love to do is also to look at developmental origins of health and disease, look at exposures, and how they affect humans with time and so on. So I'm going to stop there for now. I'd be happy to take questions. I want to acknowledge my wonderful lab. Wian and Esther are here today. They're amazing. We call ourselves a Girl Powers Lab. We do, we divide and conquer, and we've been able to do all this also with the help of our medical and undergraduate students. I want to thank our environmental liver disease group at the CTRB. I want to thank my KO1 mentorship team. One of them, Kiari, is here, who has been an amazing supportive mentor. I appreciate you. My collaborators, as well as the different centers and people that help me with startup and helping me be a successful early stage investigator. And so with that, I'd like to thank everybody. And that's our website if you want to know more about our work. And I'm supposed to show this. So yeah, thank you. So any questions?
What a tour de force, Banrida. You have so many things that you're working on, and it's very exciting, and certainly, I think I asked questions as we went along. I am curious if you've mentioned microbiome differences and so forth. Have you also looked? Well, one specific question. In terms of are you measuring the amount of food that the mice with exposure? Are they eating the same amount of
So we do routinely measure food consumption, and we do the metabolic cages too. It's another layer of measurement. We haven't really seen any differences. But I do want to add that it has been hard to measure food consumption with our females because they're generally active. They play with the food, the high fat content crumbles the food, and then we end up not getting good measurement. So, but with our metabolic cages study, we haven't really seen any difference yet.
I was also curious, you mentioned that the female mice with exposure and the 12-week exposure showed decreased activity in their cages, and I wondered if you had looked at thyroid hormone levels in those mice.
We have not. No, we did look at our acute PCB study. That there was just a lot of variability with the hormone levels, and we don't have the best technique to look at those circulating hormone levels in mice. Yes, but it's something that we definitely want to, especially since we know that the thyroid receptor is another target. So, yeah.
Yeah, now that the thyroid hormone receptor is a target. So, but you look at thyroid hormone action in the liver.
Okay.
It's regulated by thyroid hormone.
Yeah, we have not done that, but I think we could do that. Yes. Yeah, but when we're able to measure those. I think there were obviously sex differences in T3 and T4 levels in the mice that we were measuring them, but we saw some non-significant trends. But I think that variability just it was just hard to believe it or not. You know.
Sample size.
Yeah, and we're still looking for better kits or better ways to do that. So, yeah. All right, I don't.
Are there any positive effects that are there you also see?
Unfortunately, I mean, not really. So we did see positive effects with our males. They seem to be less, less susceptible, at least to the steatotic effects, because they were just too. I think part of it was they were also so fat anyways, which is why we were, be changing our exposure paradigm and our feeding paradigm. But we didn't really see any positive effects. I mean, sometimes we thought we saw positive effects because we're like, there's no changes in cytokine levels, if you will. But if you dig further, especially with our dioxin-like chemicals, it actually seems to be more of an immunosuppressive effect. So, yeah.
Let's see if there are any questions.
Looking, I don't think, okay, sorry. Okay, there's a question. Thanks, Dr. Wahlang, for an excellent review of environmental factors influencing hormonal levels. Besides elevated liver enzymes, which biomarkers correlate with environmental effects on hormonal imbalances that can be used routinely? Okay. Thanks for the question. As of now, we don't have, we're still working with these, especially with our females. So we don't really have, a clear answer to what specific liver enzymes could be used, but we are currently working on the ACHS study to actually identify what liver enzymes could be used as potential exposure biomarkers. And then another question is, serum testosterone levels seem very high for mice. Could we also use LCMS method for that? We could, yes, we could. But again, we have measured these testosterone levels using different methods. One is with an ELISA kit and one is with a Luminex, which is a high throughput ELISA B technology. And we're still seeing those effects. So I think it is a real effect. And we have look at those levels also in other C57 black 6 mice of the same age. And they, at least the control mice, they seem to match. So we think that it couldn't be a method effect. All right. So there are two questions. Sorry.
Yeah, what's going on in the pituitary?
So what about fibrosis markers?
We have not seen any evidence of fibrosis with these studies that we have right now. We've seen with a very long-term study that we've done, not very long-term, but it was a 22-week study, and we've seen that only one time. So we're a little hesitant to say that this is actually an effect. But we're still, that's why we're prolonging the duration of, not the duration of exposure, but like the duration of the aging out the mice to actually see if we're missing those effects with the 12 weeks. Yeah. And I believe in some of our population studies, not NHANES, I think it was again with the ACHS. This is also unpublished data that we have, where we group these chemicals together and look at the toxic equivalency quotient, which is the toxicity effect based compared to TCDD. And I think we did see some associations. I believe this was with the male with some fibrotic markers. I need to look into that again. So we need to get that published too. So, but that was with work done with Matt Cave and Linda Birnbaum and other collaborators. So, yeah. All right, I think we're good. Well, thank you so much.